Welcome, everyone. Good morning, good afternoon, and good evening to YPOers and friends around the world. I'm Dick Simon. I'm chair of the YPO Psychedelic Medicine for Mental Health Group. I spent all my time helping advance this work, both because of the incredible evidence and data for its efficacy, addressing otherwise treatment-resistant, really intractable mental health issues. And on a personal level, having learned in our family how difficult and intractable these problems are. Currently, I'm co-founder and CEO of Sensorium Therapeutics, which is a drug discovery company focused on translating insights from nature into transformational therapeutics for those suffering from mental health illnesses and other central nervous system conditions. I'm grateful for the involvement of many YPOers in what we're doing at Sensorium. So this call is going to be 60 minutes with question and answer session following my conversation with Mark and David. If you have any questions, please submit them using the Q&A feature on your Zoom screen, and we'll get to them during the question and answer session. And please note that this is being recorded, and the recording link will be sent out to all registrants, which is a good reason to sign up for future programs, even if the live timing doesn't work with your schedule. So we're incredibly grateful and privileged to be joined here today by Mark Russ and David Olson of Delix Therapeutics. So Delix is a neuroscience company. They're focused on harnessing the power of what they're calling psychoplastogens, which we'll get into in more detail, but neuroplasticity promoting therapeutics to better treat mental health disorders at scale. The company's compounds are capable of rapid changes or neuronal changes in targeted areas in the brain. Well, most of these are inspired by psychedelic compounds and preserve their efficacy promoting mechanisms, but avoid the hallucinogenic or trip properties and potential other safety liabilities. Delix is advancing these to bring patients FDA-approved take-home medicines that will serve several unmet needs across neuropsychiatric and other neurodegenerative diseases. They're headquartered in Massachusetts and building a world-class biotech company. Their research has been published in many of the top scientific journals in the world, and stories about them have been in The Economist, The New York Times, The Wall Street Journal, Bloomberg, Forbes, and more. So who do we have here today? Let's start with Professor David Olson. He's a co-founder of Delix. He studied chemistry and neuroscience at Stanford and at the Stanley Center in Boston, the Stanley Center for Psychiatric Research, which is at the Broad, which is a combination of MIT and Harvard activities. His academic lab now is at the University of California. There he discovered that psychedelics promote neuroplasticity in the brain or in the cortex. He there coined the term psychoplastogens to describe small molecules that produce rapid and lasting psychedelic and ketamine-like effects, but again, without the trip experience there. David's currently head of UC Davis Institute for Psychedelics and Neurotherapeutics, and he's the Chief Innovation Officer at Delix. Mark Russ is the CEO of Delix. He's globally experienced as a life science biotech leader, most recently served as entrepreneur in residence and advisor to one of the top biotech venture capital companies in the world, Atlas Ventures. Prior to Atlas, Mark served as group vice president of neuroscience at Shire, and over the course of his 13 years there, he led teams up to 1,000 people, helping them build a $2.5 billion neuropsychiatric business. So much welcome, Mark and David. So we're going to do this in the format of questions. First, I'll be asking questions that Mark and David will be promoting sort of a fireside conversation, but without a fireside because we're on Zoom. And then we will go about half an hour in to questions that are coming from you, from the audience. So first, to both of you, just in your own words, what kind of a company is Delix and what is it doing?
Yeah, thanks, Dick. David and I couldn't be more thrilled to be here. Hi to everyone who's dialing in. Thank you for your interest and appreciate you being here and sharing a bit of time with us in the space. Delix is, I think you touched on, Dick, our simple aspiration, it's simple to articulate, very difficult to execute on, is to be one of the world's leading neuroscience companies from 2030 on. We're getting there from a team background that Dave and I, often joke is sort of on our most optimistic days, maybe 49% optimistic, 51% jaded and cynical, sort of neurodrug developers. All of us have the scars, the victories, and the deep history in the neuropsychiatric, neurodegenerative, and a few other spaces in neuroscience over the last 20 plus years. We're very passionate about it, and not just because we've worked in it, but because, as I suspect all of you who are watching and dialing into this Zoom today are, whether it's yourself, a loved one, a friend, you can think of someone in your mind right now who needs more help, needs a different treatment, who, maybe things worked well with existing treatments for them, maybe they didn't, but you see the gaps and we can do better. Delix was founded on the premise that this psychoplastogen platform that we're happy to speak to further on this call offers a new way, a new common thread, a new understanding of how not just Delix, but many companies and many approaches can work to better treat mental health disorders at scale. And specifically for those of us who spent this past 20 years working on medicines that may mask, mitigate, or otherwise address symptoms, offer the chance to actually address underlying disease pathophysiology. So in other words, take steps towards words like healing, repairing. And that's what we're really excited to do. And we're happy to tell you more about today.
Yeah, just to echo what Mark says, I think that really at the core essence of Delix is this idea of harnessing neuroplasticity to heal damaged neural circuitry, to produce kind of lasting changes, long-lasting changes after just one or a few doses of a drug, which I really think is a paradigm shift in the way we think about trying to treat mental health conditions.
Great. Can give a sense, what brought you together, with your different backgrounds to start and build Delix?
Yeah, so I can start and just kind of talk a little bit about where the company came from. I've spent my career focused on this idea of using compound-induced neuroplasticity to treat brain conditions. And it was, probably back when I was a postdoc at the Stanley Center, and this was a time in neuropsychiatry when everyone was getting very excited about ketamine. It was before ketamine was an FDA-approved treatment for depression. But it was also a time when there was a lot of preclinical work to understand the mechanism of action of ketamine. And there was a lot of evidence from people like Ron Duman and Lisa Mantesia and others that suggested that ketamine might be working by promoting the growth of really important neurons in a part of the brain called the prefrontal cortex. And when I left the Broad to start my independent career at UC Davis, I was very interested in trying to identify ketamine-like compounds in terms of their effects on structural neuroplasticity, but that maybe had some advantages. Maybe they were more efficacious or had better safety profiles or might be more scalable. And that really led us to start studying classic serotonergic psychedelics, and eventually to pivot into non-hallucinogenic variants. So my background is actually in both chemistry and neuroscience. And so after we've made some of these early neuroscience discoveries, we were able to start tweaking the chemical structures of these molecules to produce these non-hallucinogenic variants. And I guess, when we were building the company, we were looking for the leaders in the space who could really elevate Delix to produce a world-class neuroscience organization. And that's, what I linked up with Mark.
Yeah, thanks, Dave. Look, Dick, the first and simple answer to when we first crossed paths and shared a passion for the space, the first thing that really brought us together was, 100%. This is the story, not revisionist or anything else. I asked, the first thing I ever said to Dave was, heard about the research, heard about what you're trying to do. I think you heard a bit about what I'm interested in. Tell me, Dave, it's the year 2030. What is Delix to you? Right. And what you described and what we talked about on the Zoom, first thing we ever heard from him was exactly that same shared aligned vision. And that holds true for most Delix team members here. We're excited by the space. We're excited by what we're doing. We're also excited about working together and building something, building a team, building a culture, and ultimately making an impact for the people we care about in our lives, and obviously all the others out there that numerically we understand need more treatments and better help. So there was a wonderful alignment from very early on about where we want to go. Personally, though, my aperture in the space came from the other direction. Dave started from the very early kind of scientific sense of looking at ketamine and why it was a potent psychoplastogen and what else could work. I came from the commercial side originally. So I worked for many years in US, Canada, Europe, Japan, around the world, launching and commercializing a range of neuropsychiatric and other medicines. So I saw firsthand, what it takes, regardless of what the data, regardless of whether a drug is approved or not, is what it takes to get it paid for by insurers, a prescriber to change habits, a patient to stay compliant, a policymaker to be motivated to achieve the change necessary in a regulatory or other system to bring something forward. Basically, how make something accessible. In 2016 or 2017, or about then, the company that Shire, the neuroscience business I was leading, we'd grown very big as a company at that stage. We had a big rare disease business, we had other businesses, and we decided we wanted to spin out our neuroscience business and create a separately listed entity. So this is all kind of publicly available information. We announced it years ago at JP Morgan at a healthcare conference. And that was a time when Pfizer, Lilly, all these global titan pharma companies and neuro companies were exiting the neuro space. They were shutting down their neuro divisions. They were shutting down their research divisions because of how difficult it was, because we were still working with SSRIs, antidepressants, with the same kind of approach that had been taken for 20 years. It wasn't for a lack of trying. A lot of dollars, a lot of effort, a lot of time from some of the world's most brilliant people was spent in this space, but it's very difficult to drug the brain. It's very difficult to find new modalities, and we were stuck with the same old. So some of these companies exited. We'd planned to spin out this neuro business and kind of zig in while others were zagging out. And when we did that, I was tasked with thinking, okay, well, what else is out there? What is next? What's looking around the horizons? If you're going to build a neuro company, what are the most promising areas of research? For those of you who may be in the space or exposed to it, if you've heard of a company called GW, that in the endocannabinoid pathway, different from our topic today, but that was a time in where there was a lot of promise in that approach. Few other approaches in shorter acting or faster acting antidepressants using different mechanisms was being considered. What Dave talked about with ketamine, or in the case here, a company called Janssen taking that S-ketamine variant through approval. And it was the beginning of what I call the sort of the Wild West initial psychedelic science days, where wonderful researchers at Imperial College, Johns Hopkins, many just brilliant folks that we're all standing on the shoulders of their research and work. The MAPS folks, Rick Doblin and others who've been working for 20 plus years, some of them longer, to demystify, destigmatize, and improve the understanding of the psychedelic science space. It was really starting to come together. And to me, my kind of point of entry was, it was inconceivable that these sort of rapid and sustained effects were not somehow attributable. Maybe it was 10%, maybe it was 99%, maybe it was somewhere in between, but somehow attributable to rapid structural and functional neuroplasticity changes, just from my background in the space. I was curious about that. There wasn't enough data, there wasn't enough talk about it, but there was something there. What also was there to me, and we can maybe talk about this later, is from a scalability perspective, I didn't quite see the path to these potential treatments being what will change the course of mental health care for tens of millions of patients. I saw the path to thousands, tens of thousands of patients. But at that point, it wasn't clear to me that the scalability was there, but wonderfully promising. So I had been fascinated by it, stayed close to it, tracked the research, paid attention as the first generation companies started getting approved, launched, and the regulatory path was being laid clear in front. And it wasn't until though I met David and the founding kind of scientific team that I said, this is it. This is the marriage of both the promising science, the promising potential for this new modality, combined with scalability and cost, which we can talk about.
Great. So let's sort of jump into that for a moment. There are a number of classic or first generation, what you may refer to as first generation or others as classic psychedelics. Why isn't LSD, psilocybin, MDMA, and others kind of the answer that's being sought? They're not SSRIs. They seem to have very, very promising trial results. Why not more of that?
Yeah, I mean, I'll sort of jump in there. First, let me just be clear. We don't believe that this is an either or story. It's not about you don't need the trip, and it's not about you definitely do need the trip, in our opinion. We think the world needs more brain health companies, not less. And, we're cheering on everyone in the space who's got the kind of fortitude to do what it takes to get a new potential therapeutic approved for future benefit. In fact, the wonderful people and teams who are leading and will soon see some of the first approvals in this first and second generation psychedelic science space coming forward. I mean, that'll be great for everyone. So some of those companies, in fact, have even invested in us early on. So we're very close. We understand that you need both approaches. For us, however, and I think not just us, I think it's well understood in the literature and growing in understanding in the literature, there will always be a significant portion of people for whom a first generation in-depth classic, as you said, multi-hour experience with therapists, with onboarding, with offboarding, with everything that goes into that will not be feasible based on a range of factors: cost, complexity, time, something called comorbidities that we could speak about further. So having a pre-existing condition such as, not just for yourself, but in your family lineage, schizophrenia, bipolar disorder, psychosis, a history of these things in your own family history would actually be contraindicated for many of these first generation or second generation treatments. So those are some of the factors. But the simplest way I tend to think about it, Dick, is in all the years of sort of launching neuroscience or neuropsychiatric, neurodegenerative medicines around the world, I kind of think about it in the 5 Ps. Does your medicine have a chance to make a difference in order of priority against 5 things? First, patients. That's the most important P. Will what you are developing lead to a meaningful difference versus the standard of care today for patients? If it's obvious, but it's interesting sometimes how that can be forgotten. That really has to be the North Star. If this gets to the finish line, is this a meaningful difference versus existing options today for a patient? That's number 1. Number 2, payers. Will payers pay for this? Or does it require payers, by the way, for those on the line, I would define like insurance companies here in the US, around the world in single payer systems, national healthcare systems. Will they deem this new intervention cost effective and worthy of payment? It's, by far the number 1 factor in a future medicine success is how quickly or not that reimbursement occurs, by far. So that's number 2. Number 3, prescribers. Is what you are offering complicated for a prescriber to get their mind around and administer and change patients from one treatment to another or roll it out? Or is it easy? Easier, better than harder. So prescribers, number 3. Number 4, pharmacists, psychologists, the kind of care network that surrounds a patient. Is this new intervention, is this new medicine something that makes their lives easier or harder? And there's many factors that go into that, but that's kind of fourth on the list. And then finally, policymakers. Is this new intervention, this new medicine, is this something that from a public policy perspective is highly motivating? Is it challenging? Is it what does it mean if you're a healthcare policymaker or even a broader policymaker? And in those five metrics, a non-hallucinogenic take-home medicine that can sit on your pharmacy shelf, be dispensed from the pharmacy, sit on your medicine cabinet at home, and be self-administered, which we can talk about more, hits many more of those metrics than one that does not.
Yeah, so just to kind of follow up on, oh, did I lose you all? Are you still there?
No.
My internet is acting up a little bit. So just to follow up on what Mark was saying in terms of, why these first generation compounds are just not maybe the most scalable. So first I want to emphasize that 100%, I absolutely am hopeful for the day when treatments like psilocybin and MDMA become available to patients. I do think that some people will definitely benefit from these medicines, but it's important to recognize that these are very powerful drugs, and to be administered safely, they have to be given under the care of a healthcare professional. And that obviously increases the cost and the complexity of the treatment. And when you consider the fact that 1 in 5 people will suffer from a neuropsychiatric disease at some point in their lifetime, we're talking about 1 billion people worldwide. So if we really want to make a dent in this enormous challenge, we need incredibly safe, orally bioavailable take-home medicines. And that's where I think the Delix approach can be useful. Now, everyone thinks about the hallucinogenic effects of the first generation compounds as something that limits their scalability. And Mark mentions the issues with comorbidities. And it's also important to remember that most neuropsychiatric diseases are highly heritable. There's a genetic component to this. So it's likely that if you are suffering from depression, someone in your family tree probably has a history of one of these disorders that might preclude you from actually using a hallucinogenic psychoplastogen. But the other thing to keep in mind is that these drugs had not been optimized to be medicines. And there are some other issues associated with them that people tend not to think about. Everyone thinks about the hallucinogenic effects limiting the scalability, but there are other issues: cardiotoxicity. Drug like Ibogaine binds the hERG channels in the heart, and several people have actually died while they were trying to have their substance use disorders treated during their Ibogaine therapy session. Other drugs bind to 5-HT2B receptors, which can cause cardiac valvulopathies and heart attacks. Still, other drugs like MDMA have psychostimulant properties and have some abuse potential there. So there are other factors to consider when developing new medicines, and we try to take all of those into consideration.
So how would you address the question that the flip side of all of that the psychedelic or hallucinatory effects aren't potentially key to delivering some of the benefits that are being found in a number of the trials that are being done? How can you separate the two and know that you'll still get efficacy?
I mean, that's like the number one, question that we think about, or that, obviously a company like ours get asked, to trip or not to trip. That is the question. Again, who are we to judge or preclude anyone that we may know, or experiences of those that of folks you meet who indicate in many cases these are one of the most transformative experiences they've had in their lives. That's why we need these treatments. That's why many people will benefit from them, and we hope that they will be offered to those who can afford, or the time, cost, complexity, or anything else associated with undertaking those treatments. What we're saying is, how sad would it be if that was the only way that you could access the power of psychoplastogens? Significant portions of the population would be limited from benefiting from these if the only way that time and clinical data will ultimately be the answer. But if that's the only way, we're going to drastically and dramatically under impact the population, versus an approach in which both become available and commercialized. Now, as to why we feel so strongly about this, why don't we start first with our conviction in why we believe it's possible to disaggregate the hallucinatory component through our kind of what we would call our flow scheme, our structure activity relationship understanding. Maybe speak to that first, Dave, and then we'll jump into the reasons to believe on efficacy.
Sure. So there are a variety of preclinical models in neuropsychiatry are really, really challenging. But in terms of preclinical models for predicting hallucinogenic effects in people, there's a few assays that are particularly good. And if you look at the correlation, you're seeing really, really strong correlations. And so we feel pretty confident that the molecules that we produce are not likely to produce hallucinogenic effects in people. In addition to looking at the behavioral effects, we can look at direct activation of specific circuits in the brain. And so that is doesn't require any interpretation of these behavioral type effects. So that's how we can separate the hallucinogenic from effects of the drugs. But the question really is, do you still retain the efficacy? I think that is really critical. And so, there's a number of reasons to believe that we can still do that. First and foremost, clinical data trumps everything. And there's a variety of indications from small trials of various non-hallucinogenic analogs in the literature suggesting that they may have some therapeutic potential. And they are definitively not hallucinogenic because people have taken them. For some of our novel compounds that have not been introduced to patients yet, one of the things that we do is we do direct head-to-head comparisons against the first generation compound. So we compare a Delix compound to something like ketamine or psilocybin or MDMA, and we show it has comparable efficacy, both in its rapidity and in its sustained effects. And we show that this is greater efficacy than traditional antidepressants like an SSRI or something like that.
Yeah, that's it. That's a really important point that in my career, so going back 20 years, I have not had the benefit ever of seeing preclinically. So that means in animal models before you move into human trials, direct comparison data that shows how your new medicine is behaving versus other medicines that you can draw a line out to the clinic or into human trials or even approved drugs, and then draw a line back to how they perform preclinically. Delix compounds, as David described, are matching or exceeding the performance of ketamine, psilocybin, and others on efficacy measures, not just kind of showing that they're non-hallucinatory, but on the reasons that they work. In addition to that, Dick, we won't bore your audience too much with on the details here, but there are some very directly observable, literal correlations between cortical neuronal growth that are observed in animal models and efficacy that some of our wonderful scientific advisory board members and others have published in the past. And these are, in our world, additional data points that you don't normally get preclinically. It's very hard to show something called translatability from animal models to human trials in neuroscience. It's hard to ask a rodent or other animal, dog, simian, how are you feeling, right, in a depression model? So you have to do other mechanisms, other validated tests to bear this out. And when you can add direct comparison the way we do, we have pretty high conviction by convergence of multiple reasons to believe. Yeah, I think there's, for anyone interested in that, we have more information on our website and other places about some of these studies, but we're very enthusiastic about the likelihood.
Great. We keep using the term psychoplastogens. Can you define that in a way that people will understand how the distinction?
Yeah, sure. So, in neuroscience, there's a lot of compounds that can promote neuroplasticity. When I talk about neuroplasticity, I'm specifically talking about the growth of neurons. So if you think of a neuron like a tree, you can think of its dendrites like the branches and the leaves like the synapses. So, the compounds that essentially are like Miracle Grow for neurons. The difference with a psychoplastogen compared to other compounds that promote neuroplasticity is how rapid the response is induced and how sustained. So compounds like SSRIs, they promote the growth of cortical neurons they just do so on a timescale that correlates with their clinical efficacy, which is weeks to months. So daily administration for weeks to months, you see the slow growth of cortical neurons and you see this slow rescue of disease symptoms. A cycloplastogen can do that within 24 hours. And this rapid growth seems to be sustained. And there are studies in the preclinical models demonstrating that, for instance, a single dose of psilocybin can promote the growth of those cortical neurons for at least a month, which is, I think, what differentiates a cycloplastogen from other types of plasticity promoting small molecules.
Great. In a couple of minutes, we're gonna be switching to questions that are coming in from the audience. So I'll just remind people that if you have questions, please use the Q&A function on your Zoom screen. On a layman's level, how do you create non-hallucinogenic psychedelics, or how do you make a psychoplastogen? How do you dial out the trip effect, if you will?
Sure. So small molecules are modular. They have parts of their structure that impart various types of function. And so medicinal chemists will do something that we call a structure function study. And what that simply means is we take the structure of the small molecule and we chop it up and we add pieces here and take away pieces there, and doing a whole bunch of these small modifications and seeing how that impacts the ultimate function, which is in our case, the promotion of a cortical neuron growth and the rescue of those depression symptoms. We are able to discern what aspects of the structure give rise to the beneficial effects that we're looking for, and what aspects give rise to the hallucinogenic effects. And then once you know that, you simply craft a molecule that retains what you're looking for and cuts out what you don't want.
Yeah, to that point, Dick, that's why over time, and we happy to share a little more about where Delix literally is in our drug development process. But we have over 1,500 compounds now. If you think of a funnel that are at various stages of categorization, with those at the bottom the most characterized and ready to move into human trials soon. And on a pipeline, like any drug or medicine, biotech company has a pipeline that shows, what are the assets that are in human trials or approved, and then what are ones that are at earlier stage that you're still characterizing. For Delix, when you move down that funnel, the actual structure, if you will, that a medicinal chemist would look at for these compounds, once you get one or two rows down in what's called the discovery stage, or the close to IND enabling study readiness stage, there would be no inference or interpretation that this is even associated with a psychedelic scaffold or compound that a medicinal chemist who's trained to look at these things would see. Now, that, by the way, that doesn't change the fact that all Delix compounds, regardless of what they are have all IP protection. These are all what are called novel small molecules with composition of matter, intellectual property. So they are all their distinct, new, defined, independent things. But just from to Dave's point, the way in which our excellent team has refined, learned, and progressed our understanding of how to make these compounds has actually moved beyond what one would consider to be a psychedelic inspired scaffold.
Great, thank you. Many more questions here, but I want to go to some that are coming in from people joining via the web. So, and there are several along this line. Assuming the drugs developed do in fact increase the brain's neuroplasticity, is there another component needed? Counseling, psychologists, psychiatrists, and without that, how are you going to set up a system where the new or improved neural pathways actually lead to an improved life outlook to overcome depression, anxiety, and other mental health issues? Yeah, how important are the additional?
Great question. And we should all be so lucky in our life. Forget even about mental health. I mean, pick any injury, any condition that we may suffer from in life. If you can append behavioral therapy to it, or have someone to talk to, whatever your outcome is in your in your intervention, it's going to be better if you have the good fortune to have the time, effort, and qualified therapists to spend time with. So we think it's a great thing, and we also think that more broadly, the shift towards cardiovascular health and the growth of physical fitness is now being mirrored in many respects by attention to mental health and an awareness that we're dramatically under-indexed on the amount of therapists and others who we can access, let alone afford, in our own lives. So that's a long way of saying behavioral therapy appended to any treatment is a good thing. Delix, however, will be seeking to study and understand our medicines as standalone monotherapies. In other words, we want to study in clinical trials exactly how much efficacy, exactly how much benefit, exactly how much change you get if you only take the medicine itself. You do not append behavioral health or therapist help to it at all. And the reason for that is because we believe that, first of all, this common underlying pathway of cortical or neuronal atrophy, the withering of the dendritic spines, on neurons, and the repair of them is at the heart of the efficacy of psychoplastogens. However, from a payer, from a reimbursement, from an insurance, from an individual perspective, from a comorbidity perspective, we want to make sure that if you only take this, what is the gain you get? Because that's what's going to get you get compounds on the market that can be delivered to the home, that can be taken and ingested at home, and thus compounds that, should someone choose to, you can append and attach to either an in-person behavioral health visit, if that's something that you're seeking to do for whatever neuropsychiatric condition you're facing, or with the rise of telehealth, be able to match and mirror at the home. But let's not forget as well that there's a range of conditions, schizophrenia, other indications that you start moving more towards the neurodegenerative direction, where a classic psychedelic can't even go, and where by measuring the efficacy and seeing what Delix compounds can do over time as monotherapies, we may have a place for that. Without the Delix-like approach, no treatment may work.
Great. Thanks. Can you give a sense as to, again, several questions all around this, kind of where you are now as a company? You raised a Series A year and a half ago. The plan was to move into phase 1, and just where are you, to the extent you could talk about it in an open forum like this?
Yeah, sure. So, I mean, we're headquartered in Massachusetts, just outside Boston. We've been a virtual team. We were built in COVID, and very quickly went from, I think, Dick, you were talking about fireside chat. I mean, our literal first board, or not even board, just team meetings were fireside chats in the backyard of my house on the fire pit with the first 3 or 4 team members. We're 20 large right now, full-time staff, another 15, close to 20 part-timers. And we're moving into our permanent labs and headquarters next month. We've had temporary labs in the Cambridge area over the past couple of years. We have 2 lead compounds that completed all their animal studies, toxicology studies, et cetera, over the course of 2022, and are moving into phase 1 human trials. First one this summer and the second one in the fall. Those are, very exciting, important, critical steps for any company, us as well, to begin moving into human trials. And we expect a third Delix compound to be sort of nominated to move into the final stages of preclinical studies to be ready for human trials in January of next year. So it's called IND enabling studies. Adjacent to that, I think you've referred to it, we've continued to share some of our insights and Delix sponsored research, and most recently in a journal called Science that we're happy to share. There's 2 more, a new compound that we'll publish on at some point later this year, hopefully, and then some more insights into our approach towards the end of the year to try to share what we know with the broader space and hopefully help lift the whole space up. You mentioned financing. That's right. We closed our Series A at the tail end of 22, and we're not fundraising in any way right now. We're kind of focusing on execution, doing what we need to do in our path to grow the company.
Great. So does that leave you fairly insulated from the incredibly challenging macroeconomic landscape over this last year and a half, couple of years?
I mean, more than many, but it's unavoidable that, you know, I think some analysts have referred to it as a nuclear winter for biotech over the year 2022 and, you know, through the beginnings of this year. That's certainly the case for many of our friends, and some great research, great companies, great science, but who, for whatever reason, you know, are facing some challenges in the capital markets. We're fortunate that the long-term investors that we have, you know, some of the leading biotech investors in the world, you know, supported not only the record-setting Series A for this psychedelic science or, you know, psychoplastogen space, but that they're not traders, they're long-term investors. They're folks who really share our vision that Dave and I spoke about for building a leading neuro company in 2030. So we're in it together for the long haul here, and we're fortunate to be able to focus on execution. You know, having said that, you know, we all hope that we see a turn in the markets and a bit of an opening in the crossover from public to private market sentiment to motivate some of this great peer companies in the space to get the capital they need to move forward.
Question in terms of things you're working with, you had cited, you know, risks with ibogaine for substance use disorders. Are you doing any things related to substance use or addiction?
So we technically are, in the sense that we've signed an agreement with NIDA, the National Institute on Drug Abuse, who approached us based on the promise and the early science of a couple of our compounds, actually, for substance use disorder indications. And, you know, that is a critical, both moral and strategic unmet need, but a very challenging preclinical and clinical development path to undertake for any company, let alone a smaller, you know, private Series A company like us. So we're fortunate to have been approached by NIDA and partnered with them. So they're actually funding the study for us in substance use disorders for Delix compounds.
Great. There have been a lot of conversations around the efficacy of you know, psychedelics for certain ailments, but one of the questions, and again, it's you're not doing directly psychedelics, but it's certainly relevant here. In terms of issues of consciousness, where there's shifting consciousness, like schizophrenia and the like, is part of the issue that we don't yet really understand what that means, what consciousness is about, and that we can make neurons grow more, but until we unlock some of that, there are situations that are very difficult that we won't be able to address.
I think well, go ahead.
No, you can go ahead, Mark. It's fine.
I think I understand the question, Dick. Like, is it hard to understand, like, define consciousness, right? And you know, the four of us will have, you know, potentially, you know, in isolation, if we write it down, we may have sort of like different definitions of what consciousness is, and thus, you know, a different interpretation of what the utility of introspection may offer or provide to unlock, quote unquote, benefit, you know, changes or positive changes in that introspection. I'm certainly not, I don't think that's an answer that I'm capable of giving, because we all have different, you know, positions on that. What I do know is that there's a literal visual, countable, differentiated neuronal growth change that occurs in patients with schizophrenia and versus other conditions that can, if directly repaired, changed, leads to positive outcomes. So this is more of like an objective physical healing and repair question, and not an introspective one to us.
Yeah. So what I wanted to emphasize about that is, if you look at the brains of patients who had schizophrenia, you will see gross morphological changes. There will be physical withering of key brain structures. And it's believed that this atrophy leads to kind of an imbalance in certain neural circuits. And that's what leads to some of the so-called positive symptoms of schizophrenia, which are hallucinations and other things. But it's also believed that it's linked to the negative symptoms, which are things like anhedonia and the cognitive symptoms of the disease. The goal really has been for a long time to identify medicines that could regrow those neurons to reestablish synaptic connectivity and heal those damaged neural circuits. The big issue, though, with using a drug like psilocybin or LSD for a patient with schizophrenia is this worry of exacerbating the disease, or in a patient who has not presented with schizophrenia yet, but might have a family history of actually precipitating the illness. And so the hope is that these non-hallucinogenic psychoplastogens will be able to go into conditions like schizophrenia where traditional psychedelics cannot.
Great, thank you. Shifting to how you're doing drug discovery. Are you leveraging large language models, variants of AI or generative AI for what it can potentially contribute to what you are doing?
Short answer is no. We're old school in the sense of hardcore medicinal chemistry focused on and a lot of these assays and a lot of the work we're doing is so, I'd say, sort of cutting edge or on the frontier that AI models would not have the data inputs necessary and the case history necessary to provide the appropriate advice to support it. Now, you could train, and through machine learning, train to accomplish that, but at this stage, we feel we need to build the continued hands-on flow scheme based asset or compound improvements and evolution over time before we start applying AI. Now, having said that, there are various automation approaches that we've taken to increase the throughput. So in other words, be able to have the way we do assess a compound now is much faster, more efficient, and more accurate than it was 2 years ago or it was 1 year ago. So we're consistently improving the way we do our discovery efforts. We're not at if our medicinal chemists were here, Dave, they would not ascribe to the AI approach yet for us.
No, I think that, a lot of the targets that we work on are not super amenable to the AI approach at this stage. Science really hasn't advanced to the level where that has been useful for these types of targets. And the traditional approach has been much, much more effective.
Great. Thank you. What excites you most looking at the next couple of years about what you're doing at Delix, and what are the biggest challenges that you're concerned about?
Well, I'll start, I guess. I have two things that really excite me. Number one, I'm very excited for our phase 1 clinical data. I think that, phase 1 studies are pretty boring for most companies, but for us, I think it's going to be particularly exciting because we're going to get a read on whether or not the drugs that we've created are hallucinogenic or not. And that is a pretty big milestone for the company to demonstrate that all of our preclinical work has validated in humans. The other thing I'm very excited for is the future, and that is where we're going. And so, obviously neuropsychiatry is a good place to start, but I'm really excited for moving into conditions where first generation hallucinogenic psychoplastogens can't go. We've already mentioned things like schizophrenia and bipolar disorder, but I'm also excited for the potential down the road to move into neurodegenerative conditions, because the ability to reverse some of that cortical atrophy could, I think, really be a game changer for those conditions.
Agree. Won't reiterate those two. I share the opinion on both, and the second in particular. For us, as we use the validation or use the phase 1 learnings and momentum we generate to then further widen the pipeline and move into wider and wider indications outside of the neuropsychiatric side. All I'll add to that, Dick, is I'm really excited by the company and culture that we're building here, and obviously the impact that comes on the other side. We were really fortunate, last year, and it was sort of humbling or surprising that we found ourselves on stage with Pfizer, receiving something from the Healthcare Businesswomen's Association award that spoke to just where Delix ended up in terms of the advancement, the commitment, and the engagement for female leadership. at our C-suite and across the board for the broader, the broader talent base that we have. I say it was surprising because we didn't apply for this. It came from a different source. So it was surprising when it came, and it wasn't even intentional. We just kind of ended up there. But like culture, as you know, I mean, all of you on this call are leaders of various teams across different industries, and as exciting as the science is, as exciting as the promise is, the unmet needs, the markets are, people are what make the difference. And every day we have more like challenges to overcome and opportunities to seize than we have the time and people to do it. And it's really exciting to try to do that and see the incredibly talented and experienced people who are, we're fortunate enough to have joined Delix or want to join Delix. So I think the more like as that grows and as we're lucky to work with more great people and hopefully focus on keeping that culture alive, that's just something I know we all enjoy when things are going well. It's not just about the results. It's like how you get there as a team.
Great. I just want to make sure that people leave with a clear sense as to why is neuroplasticity always a good thing? Can you like get neurons growing and create a new set of problems or exacerbate? And then what's the role of structural plasticity in brain health? How do you know which direction?
Sure. Neuroplasticity is not always a good thing. It really needs to be targeted and circuit specific. So if you promote neuroplasticity in maladaptive circuits, that can lead to adverse behavioral effects. So for instance, addictive drugs promote plasticity in, addiction circuitry. But the compounds that we are looking for are more selective in the circuits that they promote plasticity, particularly the circuits that lead to symptoms relevant to depression, anxiety disorder, substance use disorders, et cetera. And so in terms of the role of cortical atrophy, though, this is something that I think is underappreciated, is that how widespread cortical atrophy is for many, many, many neuropsychiatric diseases. And one hypothesis for why compounds like psychedelics seem to have this panacea-like property, they work across indications, they work for depression, PTSD, substance use disorder, is because they're all targeting this really critical hub in the brain, the prefrontal cortex, that does atrophy in all of these conditions. And so the ability to rescue that atrophy, I think, can have really broad-reaching ramifications for the treatment of brain conditions.
Great. Do you see this ever being done as an adjunct in combination with ketamine therapy or psychedelic therapy, or are you looking more at that's one route and that has a lot of potential benefits, and you're just on a different route and path?
We absolutely see promise down the road in a patient who is going for their once yearly, twice yearly, quarterly inpatient hallucinatory psychedelic experience with therapy, with onboarding and offboarding and whatever else they may seek to do, to have what's called a maintenance of effect, or improve the maintenance in between those visits with a psychoplastogen such as a Delix one in between. Time and data will have to kind of bear that out, but that's an area we see promising. So that's not necessarily immediately adjacent to the ketamine treatment in your example. It would be concurrent with or immediately after.
Great. What about other neurodegenerative diseases like Parkinson's or Alzheimer's?
There's lots of reasons to believe. For us, the reason we didn't start there is the length of time required, the size of patient trials, the cost associated. It's just not conducive for a company of Delix's size or capital base at the start to pursue. There are other indications that make sense to begin, but over time we see ourselves moving in that direction.
To elaborate on that, is there some reason that neurodegenerative diseases are more expensive or more complicated than mental health conditions, or
the trials, the size and complexity of the trials in order, and the length of time, patient recruitment, there's a lot inherent to it that it's a critical moral, strategic, financial, imperative to pursue. Lilly, Biogen, Eisai, like these companies, when they were pursuing trials in this space, these were 4 or 5 years long, hundreds of millions of dollars, and not just one, like multiple trials necessary. So it takes it'll take a little bit of time, but we're excited to go in those directions.
Do you ever see a time when the possibilities that these drugs can actually prevent brain atrophy and are all part of advancing longevity or advancing healthy longevity from a brain standpoint?
It's interesting. We get approached often by what I would call the longevity ecosystem of investors and philanthropists and others. I think we certainly want to generate a little more data and take a few more steps up the staircase first. But there are many reasons to believe that cortical and neuronal atrophy doesn't just sit at the heart of a range of neuropsychiatric and neurodegenerative conditions that are induced by either outside in stress, trauma, acute or chronic, but inside out genetic predisposition or genetic conditions, and as such, a compound that can repair and restore. This isn't, remember, aberrant neuroplasticity. This isn't sprouting of new dendritic branches that weren't there before in areas of the brain that can cause maybe less desirable outcomes. This is repair, healing of withered or degraded areas. And with that being the case, there's a lot of reasons to believe there. But again, one thing at a time for us to be able to execute. I don't know, Dave, if you'd add anything else there.
No, I think that sounds good.
Great. Well, as we come to the top of the hour, first I want to thank you both, but ask each of you, if there's one thought that you want to make sure that people in this audience leave with, what might that be?
So I'll jump on one, maybe two things. So the first is I'm scanning some of the questions as well. I thank you so much for them. I appreciate all of them. Sorry we couldn't get to all of them yet. I did see a theme about cost and whether pursuing, I think the question is pursuing the medicalization approach may be limiting access. We believe it's actually the inverse. Compounds such as Delix's are what will be approved on Medicaid, for Medicare patients, for others, or at least stand a much higher chance of approval and access for I wouldn't call it marginalized communities, but communities with less access or less ability or capability to pay out of pocket. It's the kind of medical salon or cash pay out of pocket system that, there's nothing wrong with that. It's starting today and will continue to grow. That's actually the non-medicalized, or at least the non pharmacy dispense system is the one that is actually causing significant cost right now, or cost burdens for marginalized communities. And that's if we're choosing an approval route that goes the FDA route. Recreational, that's a whole different thing. That's a different kettle of fish. But I think it's important to state that we feel very strongly that we're improving access here with these compounds is something that is about cost, not just about science. And we feel and hope that our research will allow many more people to benefit from these psychoplastogens than otherwise would. But other than that, I think we're just, for this group, I couldn't be more appreciative of the time you spent with us. And, please reach out to Delix on our website if you'd like to hear more from any of us about what we're doing.
David.
I just echo what Mark said. I think that for me, developing these non-hallucinogenic psychoplastogens is really about increasing accessibility to as many patients as possible. We all appreciate just the sheer massive number of people who are affected by mental health conditions. And I think about, people like my parents who live in a rural community that are hours from a metropolitan area, not next to a psilocybin or ketamine clinic, that have very traditional insurance. I want those types of people to have access to really, really quality medicines for improving their brain health.
Great. Well, again, thank you, Mark. Thank you, David, so much for your time today and the amazing work you're doing in the world. And thanks to everyone who joined and for your great questions and your interest. Before concluding, for those who are YPOers, if you are a YPO member or spouse and are interested in staying informed, please join the YPO Psychedelic Medicine for Mental Health group, and you'll learn about chapter events, international events, and upcoming webinars. Final reminder that this call has been recorded, and everyone will be receiving a link shortly. Have a great day, and thank you so much.